Dr. Hansen here again. Earlier I talked about ATP in quite a bit of detail, and I do hope that you have a good feel for why those phosphate bonds in ATP are so high in energy and why breaking those bonds can release energy which can be used to drive biological reactions. Now I want to look at some other coenzymes, and in particular, I want to look at coenzymes which are built out of vitamin B precursors. So to begin with, I'd like you to categorize each of the vitamins as fat or water soluble. I want you to be able to give some idea of the chemical role that each of those 8 B vitamins plays in chemical catalysis. And then I want to zoom in on 2 of the most important coenzymes, which are built from vitamin precursors, NAD and FAD, and I want you to describe in detail how they catalyze oxidation reduction reactions. That is, given a reaction involving NAD or FAD, you should be able to identify the substrates, products, the nature of the nucleophilic attack mechanism, what is low in energy, what is high in energy, what is oxidized, and what is reduced. The most centrally important coenzymes are actually built from vitamin precursors. And obviously, as a physician, you know vitamins are going to be very clinically relevant, and you're going to need to have a detailed understanding of all the vitamins. For the sake of today's talk, though, I want to think on a broader scale, and that is that some vitamins are water-soluble and others are fat-soluble. Now, that's fairly straightforward. You should know that the vitamin C and the various B vitamins, so it's only B and C, are water-soluble, whereas A, D, E, and K are fat-soluble. And to me, that looks a little jumbled. That's because vitamins were originally called A, B, C, D, E, F, G, H, I, J, K, based upon the order of discovery. And then later, it was found that, for example, the B vitamins could be divided into a wide group of different types of vitamins. Of course, to make sense of fat-soluble and water-soluble, we kind of have to look at their structures. These are the least important of all the structures you'll be looking at in all my talks. But look at these and see if you think some are kind of outliers that are really greasy and that you don't think that could dissolve in water, but could readily go into some grease in a pan or skillet or something. Please go ahead and do that, but I need to move on. Well, as a general rule, to really be fat-soluble, probably 3/4 of the molecule has to be hydrocarbon. Do you see any examples of that? If even 1/4 of the molecule is oxygens or nitrogens, then those are going to interact with water and pull the thing in. Of course, that means like something like a fatty acid or cholesterol is going to be greasy. And here I think it's pretty clear, I hope you try to at least look at it and think about it, that I put all the hydrophobic ones on the left here. Look at this. Vitamin A, a single hydroxyl with around 20 carbon atoms. Vitamin D, a single hydroxyl, again, I think around 25, probably around 30 carbon atoms. Vitamin E, a single ether link in hydroxyl, very greasy. And again, 2 ketones in all grease. All the rest of these, they're about equal. If you're half oxygen, half nitrogen, half hydrocarbon, well, as a general rule, you're water soluble. And again, that's all of the B vitamins, which are at the center of metabolism, along with vitamin C. But again, the B and C vitamins are water soluble, and I want to focus today on the B vitamins, which are the coenzymes at the center of metabolism and which underlie a number of diseases and syndromes. In fact, these B vitamins are also the very vitamins that will be in a multivitamin that you might take for yourself or you might give to your child, such as thiamine, riboflavin, niacin, pyridoxine, folic acid, cobalamin, biotin, and pantothenic acid. One word of advice: most likely if you take a simple multivitamin such as one of these, it won't harm you. But you really should avoid megavitamins, and also you should avoid large doses of any single vitamin unless you have a clear understanding of a very specific problem and why that vitamin is going to help. Because in many cases, excessive doses of vitamins have been harmful. I want to begin with the 8 common B vitamins because they are used as precursors to make coenzymes. And those coenzymes are going to be at the center of metabolism and biosynthesis, so I'll be talking a lot of them in the context of human health. As you're thinking about these vitamins as a physician, it will help if you begin by simply looking at the names and saying, what is the clue to the corresponding coenzyme? Don't feel like this is essential, but take a quick stab and say, wow, there are clues in this nomenclature. Here, by coloring, I've given you some clues which I think might help in filling in some of these blanks. But I'm going to go on, and let's begin with B1 thiamine. Now, I'm telling you that that name gives you a clue that there are 2 atoms in there at least. It turns out to be a heterocycle that has a sulfur and an amine, has a sulfur and a nitrogen in it. Thiamine is a clue to the corresponding coenzyme because it's thiamine pyrophosphate. You phosphorylate this, it gives a big negative charge to it, and now it'll bind to the corresponding enzymes that need it. This coenzyme is essential to the link between glycolysis and the citric acid cycle, as we'll talk about in the beginning of the semester. How about riboflavin? Do you get anything out of there? There's a sugar in it. What's that? Yeah, ribose. Flavin is an aromatic base. Flavin is in flavin adenine dinucleotide, and this is a profoundly important enzyme involved in providing reducing power, as we'll talk about, and you've probably heard of FAD. If flavin was a clue, how about niacin? It's going to be similar. It's an aromatic base like flavin. How about nicotine amide, adenine dinucleotide, NAD, NADH? FAD and NADH are critical to glycolysis and to the citric acid cycle, and then they dump their electrons into the electron transport chain. These are at the center of metabolism. How about pantothenate? Now this one's a little bit more difficult, but the 8 ending tells you there's an acid in it, and in particular it's a carboxylic acid. The word pan gives you a clue because pan is in pantheism or as in Pangea, all earth, all is God. Pan means everything. And pantothenic acid is so central to metabolism that it's found almost everywhere, hence pantothenate, used as a precursor to make coenzyme adenine. When you link the pantothenic acid group to an ATP hydrolyzing one of the phosphates, you make coenzyme A. How about pyridoxine? Now, this one I don't think you're going to know. I hope you didn't bang your head against the wall thinking about it. But pyridoxine is based upon the organic nomenclature pyridine, which is simply a heterocyclic aromatic 6-membered ring that has nitrogen atoms in it. And not real complicated, pyridoxine is the precursor to make pyridoxal 5-phosphate. We're going to talk about the structures of these later, which will also help with the nomenclature. But right now I'm saying a lot of them you can kind of look at the name and get a clue. Biotin, I don't think I have a clue. I'm not sure why that's called biotin, but it donates a carboxylic acid group in a number of reactions. How about B9 folic acid? Well, the 8 group says that it contains an acid, as in pantothenate. In fol, guess what type of food this is found in? It's found in green leafy vegetables or foliage. And what is folate used to make? What important coenzyme? How about tetrahydrofolate, a very important vitamin. Cobalamin again, well, the clue is it contains the metal, cobalt, and again, it's just cobalamin, vitamin B12. Now that we've defined the names of these vitamins, if we look at the first 4, B1, 2, 3, and 5, or if we look at the last 4, B6, 7, 9, and 12, you might say, didn't these people know how to count? Well, the reasons the numbers aren't continuous is that as they were sorting out what was in what they called a B vitamin, they found that some of these weren't vitamins. For example, not a learning objective, B4 was originally just the base adenine. But of course, your body is capable to make plenty of adenine, so it's no longer considered a vitamin. In some of the other vitamins, well, they're healthy to have and stuff, but these are the 8 common ones. The numbering goes from 1 to 12. I'm going to focus mostly on the common nomenclature like you'd see on a vitamin bottle or a patient might ask you. Of the common 8 B vitamins, I like to divide them into 2 groups, the first 4 and then the second 4. Based upon what you know about FAD, NAD, biotin, folates, and all, can you categorize these two? Although there's some overlap, the first 4 are central to some types of metabolic pathways, and the second 4 are central to a different type of metabolic pathway. Can you identify those pathways? Well, in particular, what I'm looking for is the fact that thiamine, riboflavin, niacin, and pantothenate are central to glycolysis in the citric acid cycle and oxidative phosphorylation, so they're at the center of energy metabolism. And anytime I or any other biochemist talks about energy metabolism, we're really talking about catabolism, the oxidation or breaking down of fuel molecules like sugars and fats in order to make ATP. So again, anyone says energy metabolism thinking, we're making ATP. How about B6 through B12, the last 4 B vitamins? The first 4 B vitamins are central to catabolism. Not surprising that the second 4 B vitamins would be central to anabolism. That is, the last 4 B vitamins, B6, 7, 9, and 12, are used in anabolic reactions, biosynthesis, reactions which take simpler compounds and link them together into more complicated compounds using energy from ATP, NADPH, and other high-energy coenzymes. Another way to see these B vitamins is based upon their specific role in catalysis. For example, TTP either donates or accepts an acetyl group. FAD either accepts or donates a hydrogen atom. NAD either accepts or donates a hydride ion. So what's most important about riboflavin and niacin is that their corresponding coenzymes are involved in oxidation reduction reactions. Coenzyme A either donates or accepts an acetyl group. And then in the biosynthetic B vitamins, pyridoxal 5-phosphate either donates or accepts a nitrogen atom, biotin either donates or accepts a carbon atom, tetrahydrofolate either donates or accepts a carbon atom, and cobalamin either donates or accepts a carbon atom. In fact, folic acid and cobalamin are very closely coordinated, and a shortage of one can affect the activity of another. An alternative way to show these vitamins and coenzymes is in light of the central metabolics pathway chart. Now, many of you have seen this before. If you haven't, don't worry. Absolutely do not try to memorize all these arrows and everything, but we will be using this throughout year 1 and 2 in order to put everything in perspective, so I think halfway through the year you'll have a good understanding of it. For example, at the very center is this pathway glycolysis, sugar breakdown, where glucose gets oxidized one step at a time to make pyruvate. So that's sort of at the center of the figure. What I want you to do now is get a generalized feel for the overall layout of the figure. For example, starting at the very top of the diagram, all of the blue text represents carbohydrate metabolism. That is, sugar metabolism, carbohydrate, carbon, hydrogen, oxygen, molecules like sugars. And in brown on the right side of the figure is lipid metabolism, largely hydrocarbons, molecules with hydrogens and carbons, not so much oxygen, although they may have a carboxylic acid at the end of a long hydrocarbon chain. And On the left of the figure, shown in green, are amino acids. Amino acids are compounds that contain an amine and an acid. So they're kind of like carbohydrates, except they have an additional amine group. At the very center of this diagram is one of the most important coenzymes, coenzyme A. And of course, A represents, it's coenzyme adenine. There's an adenine in its structure. What I like about coenzyme A is it kind of shows that the citric acid cycle is where carbohydrate metabolism, lipid metabolism, and amino acid metabolism can all come together to make ATP. So the citric acid cycle is kind of where all these pathways can feed into each other, and coenzyme A is very much at the center of this intersection. Pyridoxal 5-phosphate is crucial to amine transfer reactions, so it will be at the interface between the green urea cycle and amino acids and the blue citric acid cycle or carbohydrates. Thiamine pyrophosphate, like coenzyme A, is at the interface between glycolysis and the citric acid cycle, and it also plays an important role in the pentose phosphate pathway. Riboflavin, flavin adenine dinucleotide, is deeply involved in oxidation reduction reactions such as in glycolysis, citric acid cycle, and fatty acid metabolism. NAD is involved in even more oxidation reduction reactions throughout carbohydrate metabolism, and you'll notice the pentose phosphate pathway uses an NADP instead of an NAD, and the phosphate marks NAD for use in biosynthesis. Again, we'll be talking about all of this in detail later. Biotin is needed for fatty acid synthesis. It's needed in some amino acid degradation, and it also plays a role in gluconeogenesis, carbohydrate metabolism. Throughout the entire central metabolic pathways chart, we see we need these coenzymes. What do? They transfer energy from one form into another. And whenever I talk about a cell building energy, what am I talking about it making? Please pause. We're talking about making ATP. Normally, in order to make that ATP, first you have to make the very high energy NADH, which then, using oxidative phosphorylation, will pass the high energy onto the production of ATP. Don't get bogged down in this, but let's take a look at the role of how these different coenzymes are bound by enzymes and interact with substrates to transfer energy from one form into another. Let's take a look at niacin, also known as vitamin B3. I simply require you to know it as niacin. Niacin can also be called nicotinic acid, and if the carboxylic acid group gets reduced to an amide, it can now be called niacin amide or nicotine amide. Now your vitamin bottles will list this as niacin, not as nicotinic acid. Can you tell me why? Well, a lot of people don't like the sound of nicotinic. It sounds too much like nicotine, which is structurally related to niacin. Of course, the fundamental difference between niacin and nicotine is that niacin is an essential vitamin at the center of energy metabolism, whereas nicotine is an herbicide which they use to kill a bunch of plants and humans. Of course, niacin is used as a precursor that is used to build up the coenzyme nicotine amide adenine dinucleotide. And will you interpret that name, nicotine amide adenine dinucleotide, and show me how every part of this name refers to every part of the structural figure? It's definitely in your interest to do that to gain an insight into thinking about vitamins and coenzymes. Let's take a look at it. Nicotine amide is a base. Adenine is a base, and you already know about that from ATP. Dinucleotide. Di means two. Nucleotide means a base with a ribose with a phosphate. We have the nicotinamide nucleotide, nicotinamide, ribose, phosphate, and we have the adenine nucleotide, adenine base, ribose, phosphate. So this terminology, you should really think it through slowly because it's so powerful. Nicotinamide adenine dinucleotide. Do not just memorize niacin or NAD. Think about the structure because it'll help you in thinking about all the different contexts that adenine shows up in. This is very important to energy metabolism, and when I'm talking about energy metabolism, I'm talking about the production of ATP. In particular, glycolysis converts NAD plus to NADH. The citric acid cycle converts NAD plus to NADH, going from a low energy to a high energy form. And in oxidative phosphorylation, NADH dumps its electrons into the electron transport chain in order to power the production of ATP, and thereby NADH gets lowered in energy back to NAD+. Let's take a look at how NAD is used in energy conversion reactions. And a really good enzyme to illustrate this principle is lactate dehydrogenase. Can you interpret that name? There is a grammar to enzyme names. The first part of the name is the substrate. The second part is the verb. So, this enzyme acts upon lactate, and what does it do? It is a dehydrogenase. And by the way, this is worth spending your time to understand this mechanism because I will be talking about a whole bunch of different dehydrogenases throughout the next 2 years. And to understand how one of them works is going to make it a lot easier for you to remember this stuff. So dehydrogenase means it takes a hydrogen off of lactate. And let's take a look at that. As with all other enzymes, lactate dehydrogenase positions the substrate near the coenzyme. Let's zoom in and take a look at that. Here I'm showing the enzyme in gray with some of its side chains in yellow and in blue and in orange. I'm showing the coenzyme NAD in green, and I'm showing the substrate lactate also in green. For the fun of it, can you see the parts of the coenzyme? Nicotine, amide, adenine, dinucleotide? Well, here's the aromatic ring of the nicotine. There's its amide group, aim for nitrogen, ide for oxygen, amide, nicotine, amide. Here's the adenine group. 2 bases, 2 riboses, 2 phosphates, nicotine, amide, adenine, dinucleotide, with the aromatic ring positioned right next to the lactate so that they can exchange energy. Here I'm showing a level of detail so we can actually see how positioning a substrate next to a coenzyme chemically allows a transfer of energy. In particular, lactate dehydrogenase dehydrogenases the lactate and it puts that hydrogen onto the aromatic ring of NADH. And I want to get a little bit of nomenclature straight here. Can you tell me whether converting lactate to pyruvate is the forward reaction or whether converting pyruvate to lactate is the forward reaction? It's kind of important to understand just the underlying nomenclature, a very simple question. So in fact, the name lactate dehydrogenase says that it is the lactate that's being acted upon. So certainly this reaction can be catalyzed in either direction. It's called the forward reaction if lactate and NAD plus bind and get converted to pyruvate and NADH, and it's called the reverse reaction if pyruvate and NADH bind and form lactate and NAD+. So lactate and NAD+ would preferentially be called the substrates, and pyruvate and NADH would preferentially be called the products based upon the nomenclature. And to be clear, The lactate is being dehydrogenased. The hydrogen is coming off and we're forming a double bond to oxygen. Based on that reaction, can you tell me whether lactate or pyruvate is higher in energy and whether NAD plus or NADH is higher in energy? Please pause and answer. This is at the heart of every metabolic reaction we'll talk about for the next 2 years. What's the difference between lactate and pyruvate? There has been a hydroxyl group on one, and I'm showing this hydrogen which is gone, and now we have a doubly bonded oxygen. So lactate has a total of 1, 2, 3, 4 carbon-oxygen bonds, whereas pyruvate has 1, 2, 3, 4, 5. Pyruvate is more oxidized. Oxidized always means lower in energy. If you think about it, you start a fire, it burns because you're releasing energy and forming the extremely low energy carbon dioxide. So here we're going from a hydroxyl to a ketone, we've gone down in energy. As the electrons on the oxygen resonated down to form a double bond, that is, as lactate got oxidized, the hydride was driven onto NAD plus to make NADH, which means that NAD got reduced. Oxidation reactions drive reduction reactions. And of course, now we have the high energy hydride on this ring. And the ring would like to kick off that high energy hydride in order to form a more resonant, lower energy aromatic compound. Now, can you go ahead and draw arrows to show how the electrons on the hydrogen would move to the NAD plus to form NADH? It's like a nucleophilic attack. I'd like to point out that whenever you see arrows drawn on any organic figure, it always represents the movement of electrons. So that is, the electrons on the hydroxyl resonate down to form a double bond. They move from the oxygen to a bond between the oxygen and carbon. And of course, carbon can only have 4 bonds, so that drives off the hydride ion with its pair of electrons to form a bond to the aromatic ring. So this is simply how I want you to draw such arrows. And I want to point out that positioning is 99% of catalysis. You position it perfectly so that the electrons here are right next to the aromatic ring. But in addition to positioning the substrate and the coenzyme for the optimal reaction, lactate dehydrogenase further catalyzes the reaction by positioning this histidine next to the lactate. Can you draw an arrow to show how this histidine residue will contribute to the catalysis? Please take a stab at that. Well, the idea is that if we want the electrons on the hydroxyl to resonate down to push off the hydride leaving group, wouldn't it be neat if we made this oxygen more negatively charged? And what better way to do that than to pull the proton off? That is, the histidine side chain here acts as a base that pulls the proton off, which makes this more negatively charged, which favors the movement of the electrons into the double bond, which pushes the hydride off. So we're looking at an extremely important cofactor, NADH, a term I'll use 1,000 times over the next couple years. And we're looking at a very fundamental reaction mechanism. This is very close to a nucleophilic attack. If we treat this hydrogen atom here as if it's a hydride ion, can you show how the pair of electrons in its bond to the carbon atom could be used in a nucleophilic attack on pyruvate to drive the formation of lactate? Please take a stab at that. Anytime you draw a nucleophilic attack, there will be 2 parts to it. One will be the electrons in the double bond resonating up to form an oxyanion. The other part will be an attacking pair of electrons. So we can show the pair of electrons in the carbon-hydrogen bond being driven off in order to reform the aromatic ring, and they can nucleophilically attack the nucleus of the carbon atom, which is doubly bonded to the oxygen. The electrons in that double bond can resonate up onto the oxygen to form an oxyanion. How is this histidine residue going to contribute to this reverse reaction? Can you draw an arrow to show that? Well, we're offering a proton here. How about we simply show that one of the pairs of electrons on the oxyanion can favor interaction with a positive charge, and again, we're simply doing the reverse reaction to now reform the hydroxyl group. Please do not memorize every arrow in every figure, but the idea that a pair of electrons can attack a nucleus and one of the bonds in a double bond can resonate up to form an oxyanion is crucial to all of the chemistry I'll talk about all year. Now, it's kind of nice to know, but you don't need to know. Take a look at this and just see if it makes sense. As the electrons on the hydride attack the ring and get added, well, the double bond has to be broken. But this is an aromatic ring. That double bond can resonate over here, which forces this double bond to resonate up onto the nitrogen, which leaves a lone pair of electrons. And that's what's so nice about these heterocycles, is that because the nitrogen can exist either resonating in the ring or with a lone pair of electrons, these aromatic rings are great in these types of hydride or hydrogen atom reactions in metabolism. And I just want to point out, you don't need to understand every detail of this, but hopefully you're getting a feel for a nucleophilic attack, and hopefully you understand how central the vitamin niacin in the form of nicotinamide adenine dinucleotide cofactors is in metabolism. We'll be talking a lot about it. But now I want to look at another vitamin. I don't expect you to memorize that this is B2, but the name riboflavin points you where you need to go. That is going to be the cofactor that's used to make what coenzyme? Flavin adenine dinucleotide. And why is this called flavin? I don't know if you've ever heard of a flavivirus. These are viruses that turn people yellow due to jaundice. Well, flavin simply means yellow. A lot of aromatic compounds have very high energy electrons orbiting above and below the very flat plane of the ring. It turns out that the flavin ring interacts very strongly and it can absorb red light and can absorb blue light, but it can't absorb yellow light, so yellow light gets reflected off. Riboflavin is a big component of a lot of vitamin pills, and that is one of the primary reasons your urine turns yellow when you take a megavitamin. So again, can you interpret the name flavin adenine dinucleotide in terms of the structure? And you see this exactly parallels nicotinamide adenine dinucleotide. We have the flavin base, the adenine base, and the dinucleotide, base ribose phosphate, base ribose phosphate. Now what's kind of interesting is you notice how we draw ribose is a cycle here, and on NAD we draw it as cycles, but here we draw it linearly. You do not need to know this. Please don't memorize it. But just think for a minute. Why would this ribose be in a linear form and this one in a cyclic form? Let's look at the ribose and its base. Adenine is a purine, a two-ring fused system, and you'll notice this huge ring points away from the ribose rather than being rotated above it. You'd think you could rotate around the carbon-nitrogen bond and put the ring over here. Why can't you? It won't fit. This ring is too big to fit over the ring. Well, flavin has a six-membered ring on both sides of the central six-membered ring, so there's no place for that to rotate, and therefore you can't have a cyclic ribose there. Rather, you have a linear one. But again, you have base ribose phosphates, a dinucleotide. Just as many dehydrogenase enzymes will grab hold of a nicotinamide adenine dinucleotide coenzyme in order to accept or donate a hydride ion, so many other dehydrogenase enzymes grab hold of flavin adenine dinucleotide in order to accept or donate hydride ions or hydrogen atoms. Between these two forms of flavin adenine dinucleotide, can you tell me which is high in energy and which is low in energy? Which is oxidized and which is reduced? I think the clue is one is called FAD and the other is called FADH2. We know that forming a bond to oxygen is an oxidation, goes down in energy because oxygen is so electronegative, the electrons fall toward the oxygen. Likewise, we know that forming a bond to hydrogen at the opposite end of the periodic table is a reduction, and therefore that goes up in energy. We look at FAD, FADH2, FADH2 is higher in energy. So FAD can accept a hydride ion to form FADH2. It will pick up the hydride ion and a proton. Also, FADH2 can donate hydride ions, or it can donate one electron at a time, which is kind of neat. However, that's as far as I want to go with flavin adenine dinucleotide and nicotinamide adenine dinucleotide, those very similarly named coenzymes which have very similar functions. And now I want to look at pantothenic acid. And again, the word pan comes into the name pantothenic because this is so widely found in so many different types of tissues. And this vitamin is used as a precursor to make coenzyme A. What does the A in coenzyme A stand for? You can see it right there, adenine. So we have the adenine ribosin phosphate. We do have a nucleotide here. You'll notice this is not a dinucleotide as in FAD or NAD. This is a mononucleotide simply called coenzyme adenine, where a pantothenic acid extender arm has been attached to the ATP. And in addition, a sulfhydryl group has been added to the pantothenic acid. I'm hoping with all of these that you are not looking at every single atom. For example, you do need to know that pantothenic acid is an important enzyme precursor that's used to make coenzyme A. You certainly don't need to know its structure. If you gave me a blank piece of paper, I do not think I could draw pantothenic acid. You probably should know that it has an acidic group. Of course, that acidic group was here, and it was nucleophilically attacked by an amine in order to link the sulfhydryl group onto it. So now it's in the form of an amide in coenzyme A. But in the form of a vitamin, this was an acidic group. Not an important detail. What is important is to recognize from the word pan, everything. This coenzyme A is at the center of a huge amount of metabolism, both energy metabolism to make ATP, as well as biosynthesis to make fatty acids and cholesterol. Very important pantothenic acid. And what's so convenient about it, kind of like with ATP, you have that huge greasy base which will want to stick to a greasy pocket in an enzyme. And you have a hydrogen bonding group with some negatively charged phosphate groups. So on the same enzyme, you'll have a positively charged pocket to help grab hold of coenzyme A and to position it to use the pantothenic acid extender arm in order to hold the business end. So when you see pantothenic acid, think, oh, something that will bind very well to an enzyme, and wow, this is like a robot arm extending out with a working end. That's the way to think about it, not every little detail. And in fact, it does have three parts. It's built from ATP, the pantothenic acid, and actually the sulfhydryl, not a learning objective, is formed out of a cysteine amino acid. And it's so simple to have the electrons on an oxygen resonate down in the carboxylic acid to pull carbon dioxide off and leave this ethane with a sulfur and nitrogen on it to use as the business end of coenzyme A. And exactly how is that sulfhydryl group used? Well, it's used to form a that's sulfur, all alcohol ester, thiol ester. These thiol esters, like a normal ester bond, are very subject to hydrolysis. This is a very high energy bond, and that means that this acetyl group, and absolutely you'll need to know what acetyl means, can be linked to and activated by coenzyme A and then it can be used to form a carbon-to-carbon bond. And we will talk a lot about this next year, have a general feel for what coenzyme A is. So again, we've looked at ATP, NADH, FADH, and coenzyme A at the center of energy metabolism. What structural feature do you think is common to each of these? Please answer. Adenosine, 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 adenosine. AAAA. Big clue. And in fact, here I'm showing those structures where I've highlighted adenosine in red. And now I'm going to move into a coenzyme that does not include adenine, and it is called thiamine. It contains at least what two atoms? Thiamine says that it has a sulfur and a nitrogen. And I want to point out that the sulfur and nitrogen of interest are in the five-membered ring. This is an unusual heterocycle used to stabilize a carbon ion, and it does transfer two carbon groups, much like coenzyme A. But thiamine, again, is what's written on the vitamin bottle. In order to get this to bind to enzymes, it needs to be modified by addition of a pyrophosphate. How could you add a pyrophosphate to thiamine? You could grab hold of ATP, thiamine, nucleophilic attack, and now you have thiamine pyrophosphate. This will bind to an enzyme in part because of the huge negatively charged pyrophosphate binding to a huge positively charged pocket in order to position the unusual heterocycle of thiamine in order to attack a carbon-to-carbon bond. That's as far as I want to go with thiamine. And now I want to look at pyridoxine. I'd like you to pause without going on and explain how the difference in these structures corresponds to the names. We have pyridoxine, pyridoxal, pyridoxamine. Please explain that nomenclature, and I'm going to move on. Pyridoxal, the hydroxyl has been converted to an aldehyde, so this could be called pyridoxaldehyde. This happens to many compounds, including vitamin A, as well as pyridoxine. They have a hydroxyl that gets oxidized from a single bond to a double bond to make an aldehyde, and it shows up in the name. This type of nomenclature will help you if you understand it rather than memorize gibberish. And of course, pyridoxamine, the aldehyde has been interconverted with an amine. This exchange between an aldehyde and an amine, or a ketone and amine, is a profoundly prevalent reaction in life. It's how we link the urea cycle to the citric acid cycle. Anytime you're transferring amines, look for pyridoxine. It is used therapeutically for people who have vitamin deficiencies or for people who have enzyme deficiencies that need pyridoxine. Less important, but interestingly, if you look at it, pyridine is simply a heterocycle which you have a aromatic, basically benzene, phenyl ring with a single nitrogen in the aromatic ring. Pyridoxine is the same compound, but now it has some added hydroxyls. If you oxidize it, pyridoxal. If you substitute amine for aldehyde, pyridoxamine. And again, look for a lot of interconversions between amines and aldehydes. In order to make this vitamin a coenzyme, you phosphorylate it to make pyridoxal 5-phosphate, and this is what will bind to many transaminases. The reason I wanted to cover these now is to give you some familiar with the nomenclature and how to think about them. When you come to AT still, I will be using these in a number of metabolic reactions as we talk about central metabolism and about metabolic diseases. If you have any questions, I always enjoy talking with you. Don't hesitate to email or phone, and I look forward to meeting you in a month. Thank you.